IBD is a serious chronic condition that can lead to debilitating symptoms and significantly impact quality of life. Now, for many patients, the journey to an effective treatment is often a trial and error, a diagnostic odyssey, and despite the growing prevalence, the causes of IBD and the path to the best treatment is unfortunately still poorly understood because of the quite intricate interplay between genetic, immune and environmental factors that our speaker will cover today. Now, awareness and research is more important than ever, and today we've got an incredible speaker, Dr. Kylie James, who will present on this topic, and which will be followed by a panel where we'll really be able to get the full picture of IBD, right from the patient perspective through to the clinic and the research. So I'm just really looking forward to a fantastic discussion today. So that really leaves it to me to introduce our speaker for today. It's a huge privilege to be able to introduce Dr. Kylie James, who heads the Gut Immunogenomics Lab here at Garvan and is really one of our faculty superstars. Her lab investigates how environmental factors shape gut immune cells and microbial landscapes. It's a bit of a mouthful, and she's going to do a fantastic job of breaking that down for us because as well as being a fantastic scientist, incredible superstar, she is also a fantastic communicator. Recently had the opportunity to interview Kylie on the Garvan podcast, Medical Minds, which I highly recommend. And of course, we'll be hearing from her directly today. So thank you so much. Please welcome Kylie.
Good morning, everyone. It's a great pleasure to speak to you today and to welcome you into Garvan to hear about the research that we do. So, as Viv mentioned, I run a lab here at Garvan, and our focus is on understanding the human intestinal tract and how it exists in health and what goes wrong in disease. So, the title of my presentation today is Getting to the Bottom of Bowel Health and Disease. So let me start here with the large bowel. I think it's a vastly underappreciated organ in the body. It's the main site of water reabsorption in the body. About 1 litre per day is taken up in our colon. It's about 5 feet long, so almost as high as I am. And the transit time of fecal material through the colon can range from 12 hours to 48 hours, and this relates to the frequency of bowel movements, so there's a great deal of variability between us. And one of the underappreciated facts of the bowel is that it's home to one of the most numerous and diverse communities of microbial species in the body as well. So what do I mean here? This is the community of bacteria, viruses, fungi that just naturally exist in our body and that are actually important, very important for normal body functions. And directly interacting with our microbiome, so this community of bacteria, is one of the most active communities of immune cells in the body as well. So even when we're healthy, our immune system is very active in our gut, and it's important for regulating the microbiome and keeping us healthy. And lastly, there's this growing appreciation of the gut as the second brain in our body. So what do I mean by this is that the gut actually has its own enteric nervous system, which directly links in with our central nervous system. So there's this communication between our gut and our brain. And there's a great wealth of research into this axis and linking neurological diseases actually also with our gut health. So the gut is very important with our general well-being. So what is one of the main diseases that you can get in your gut, and that's inflammatory bowel disease, IBD. And IBD is actually a spectrum of diseases. So the two that you may have heard of are Crohn's disease or ulcerative colitis, but there's a whole range of rarer forms of IBD, and they vary in where they affect your gut and the depth of the wall that's affected. And IBD is actually pretty common, as Viv mentioned. So about 1 in 250 Australians currently live with IBD, and these numbers are rising in developed countries. And this kind of speaks to there may be environmental factors coming into play in IBD as well. So how does IBD present? Well, it typically has these flares of symptomatic disease, which may be abdominal pain, cramping, diarrhea, bloody stool and fatigue, followed by periods of remission. And because there's no cure for IBD, we really aim to extend the periods of remission that patients are in. So taking you through the facts of IBD is all well and good, but I thought it might be helpful if we actually walk through the experience of someone with IBD. So I'm going to take you through this case study. And this lady is actually a patient of Dr. Ghali, who's with us today. So at diagnosis with IBD, she was 32 years old. She's married with one child, a full-time receptionist, so really in the height of her life. And this is quite typical that people with IBD are diagnosed in their 30s or even younger, and it's a lifelong disease. So she comes in 2009 and is diagnosed. And so she's put on a course of steroids to control the inflammation, and then a long-term immunosuppressant to suppress the immune response to calm down that inflammation. And this sees a good time of clearance of her disease. So for about 8 years, she doesn't have very serious disease. But in 2017, she's hospitalised with very severe symptoms, so extreme diarrhea and bleeding. And in that point, she's put on another drug to calm down this inflammatory flare, and then her treatment regime is changed with the addition of an immune modulator, so something to also target the immune system and extend out to remove the inflammation in her bowel. And this sees improvement later that year. But again, the following year, she's hospitalised yet again. The treatments have failed. So again, she's put on a steroid to control that flare, and then her treatment is changed completely to a different range of drugs. And this sees her in remission. So this kind of just shows this treatment odyssey that Viv mentioned that is very typical for IBD patients, because it's very much guesswork what will work for them. And often the drug will work for a time, but then fail, and then we change it, and we continually do this. So a real aim of our research is to take the guesswork out of treatment of IBD, to know what drug is going to work for patients and really give them long-term relief. Now, another really important factor of IBD, or complication that comes with IBD, is this much greater risk throughout their lifetime of progressing through to colorectal cancer. So at least twofold above the general public, and up to 6 times a greater risk of colorectal cancer. And there's a number of reasons for that increased risk, and risk factors or elements of IBD that contribute to that risk. So one is the extent of their colitis, so how much of their bowel is actually affected by inflammation. If more of the bowel is affected, they'll have a greater risk of colorectal cancer. Also, disease duration, so that risk increases the longer that they've since diagnosis. And other elements that contribute to this risk is the extent of inflammation, so how long their inflammatory flares are and how severe they are. Also, if they have comorbidities, so other diseases, and one of the main diseases that increases the risk even further is primary sclerosing cholangitis, so this causes liver fibrosis. And then also a family history, so if they've got other family members that have developed colorectal cancer, also this increases their risk. So this also adds a level of anxiety if you've got IBD to stay on top of the inflammation. So I wanted to touch on this point because I think it's also something that's often confused, is that the colorectal cancer that we see in IBD is actually different from sporadic colorectal cancer. So that's the cancer that you might get a screening kit in the mail for. And it actually has a very different mechanism from which it occurs. So the cancer that we see in IBD, so colitis-associated colorectal cancer, it comes out of inflammation in the bowel. So it has a progression through dysplasia through to adenocarcinoma. Whereas sporadic cancer actually comes from polyps. So you might go in for a colonoscopy to look if you've got polyps, which can be the early precursor for sporadic colorectal cancer. And these two cancer types actually also differ on the mutations. So these are the genetic changes that happen in the cells that can precipitate the cancer. So we don't have to go into the details there. but they also have differences in terms of their outcomes and what it means for the patients. So for colitis-associated colorectal cancer, so that linked to IBD, it's tend to be diagnosed much earlier for these patients, so at about 51 years versus 60 years for sporadic colorectal cancer. And while colitis-associated colorectal cancer only contributes 1 to 2% of all colorectal cancers, it actually causes 15% of total deaths for IBD patients. So it is a real concern for IBD patients. And because of this increased risk for IBD, it really requires much more heavy surveillance of this patient group. So they can come in for up to annual colonoscopies to look for early indications that their disease is progressing through to colorectal cancer. So with all this in mind, why do we want to research IBD? Well, because we really want to get into the mechanisms and why we see these differences in people's experiences of IBD in response to treatments. And so we aim to harness the power of genomics to increase the quality of life and the treatments of IBD patients by informing treatment choices for individual patients, and also in determining safer treatments and better ways to determine if patients are progressing down the route of cancer earlier. So what do we know about the mechanism of IBD? So we know that it's a complex disease. So what I mean by this is that there's not one individual reason or cause for IBD, but rather it results from the interaction of a number of factors. So firstly, our DNA. So this is the genetic code that we all carry and makes us unique and different, but it can also change our risk, our likelihood of developing various diseases. And we know that there's a genetic component to IBD. So if you have a direct sibling or relative with IBD, then that increases your chance. We also know that lifestyle and dietary factors can contribute to risk of IBD. So smoking, for instance, obesity, these are all risk factors. And these two factors then contribute to changes that we see in our microbiome. So remember, this is the community of bacteria, viruses, and fungi that naturally exist. So we can see in IBD patients that the diversity of this community actually decreases. And then that then has an effect on our immune system. Our immune system becomes overdrive, it becomes hyper-inflamed, and this is what ultimately causes the damaging to the gut wall. So in my lab here at the Garvan, we really try to understand people with IBD, what's going wrong, what's changing in their microbiome and in their cellular makeup. And what does this tell us about how we can change that, how we can reverse those changes? So how do we do this? Well, we work very closely with clinicians at St. Vincent's Hospital to work with patients with IBD and to access samples that we can work on. So when patients come in for their annual colonoscopies, they very generously allow us to take little tiny pinch biopsies of several locations of their bowel. And so when they do that, we record whether that tissue was actively inflamed or whether the patient was in remission, so they were responding to their treatment. And then what we do is we take those little tiny pinch biopsies of the bowel and we do a technique that's called single-cell RNA sequencing or single-cell genomics. And what this allows us to do is it allows us to look at these cells. So these are the building blocks of the bowel and the cells that contribute to inflammation. And it allows us to look at how they change compared to in a healthy individual. From paired biopsies, we also look at, we sequence the bacteria. So we look at what different types of bacteria are adherent to the bowel surface. And in that way, we can see changes in the microbiome as well. And then what we do, and what's really important for all science that we do, is we do various techniques to help us validate or verify our findings to make sure that what we're seeing and what we're reporting is true and accurate and is worth taking forward. So I thought I would take you through a little example of some of the research that we've generated and what it tells us and kind of our line of investigation. So if you remember, we take little biopsies from people with IBD, and what we do is we release the cells in those little pinch biopsies and we sequence their gene expression. And we can do that for a number of patients and thousands of cells for each little pinch biopsy, and we can pull it all together, and then we can use computers to analyze that and to draw meaning from that. So I'm showing you some of the results from this analysis here, and I think it looks quite pretty, like a nice piece of abstract work, artwork, but it's actually very meaningful. So just to explain this plot, each little tiny point on this plot, and they're really hard to see because they're tiny, represents a single cell from the gut of an IBD patient. And they're grouped according to how similar they are in terms of their gene expression or what they look like. And that gene expression is actually a list of 30,000 genes. And so here we have 90,000 cells from IBD patients, each with 30,000 cells. So here is actually represents 2.8 billion pieces of information that we can mine through, and that tells us about what's going on in the bowels of these patients with IBD. So if I take that same plot, and just as an example, I highlight one of these little groups of cells up here. So based on the gene expression, what these cells look like, we can tell that they're a type of immune cell, that they're mast cells, which are a cell that's commonly linked with allergy. So they actually are a main driver of anaphylactic shock, but they have other roles in the body, as a lot of cells do. And in this case, they seem to be contributing to inflammation. Because I've coloured these cells now according to whether they came from tissue, bowel tissue, from an individual who was in remission, in blue, or was actively inflamed, so the treatment wasn't working, they're very unwell. And that shift, that separation in the cells tells us that they're changing in this context, and they're likely contributing to the inflammation. So then what we can do is we can say, well, how are they changing? And I'm showing a plot that the details we don't need to get into, but it basically gives us a list of all the different changes in the cells in inflamed bowel versus non-inflamed bowel. And these codes represent genes that we can then drill into. So to take just one forward, we saw that this TMEM176, it's something that's expressed on the surface of the cell, and it helps it become active. And we can see that that is very high in the cells that are purple, the cells in the inflamed bowel. And we can actually look at that TMEM176 on the actual cells. So what I'm showing you here is the biopsies, and we're looking under a microscope, and we can look for the expression of these different genes by colours. And so our mast cells, those cells are in green, and when the patient has inflammation, you can really see that red colouring, that TMEM176, really come up. So these cells are turning that marker on in the context of inflammation. So it seems to be a very good marker of inflammation as well. But what are these cells actually doing? Like, we can see that they're changing, but what does that mean? So next, what we can do is we can do a range of technologies to explore what the cells are actually doing and how markers such as TMEM176 are actually contributing to that role. And one of the processes that we do here at Garvan is something that you may have heard of in recent years. It's called CRISPR-Cas9 genome editing. And the reason you might have heard about it is it won the Nobel Prize a few years ago to researchers over in America develop this technology. And what it is in very simple terms is basically they've just taken a process that naturally occurs in bacteria. So basically bacteria use this to turn off viruses that have infected them. And we've adapted that to now we can use in the lab, and so we can actually use this for our own benefit. And essentially what it allows us to do is turn off any specific gene that we're interested in. So in this example, what we can do is we can CRISPR edit our TMEM176, so this is that marker that comes up in our inflamed mast cells in IBD, and we can turn it off, and we can look at what effect that then has on the cells in terms of their survival. Do they just die? Do they change their shape, or do they change how they respond in that tissue? So it really allows us to pivot, to change cells under our own control. And we do this in a petri dish, not in people, which is very important. So we're actively exploring this technology here. Another way we can do it is therapeutically. So we can then take a drug instead and use that to turn off TMEM176 in these cells. And so we've done this here. So what we can do is we can take mast cells, which we've taken from a mouse. We can grow them in a plate and we can induce them to be activated like we see in IBD. And then what we can do, oops, sorry. Oh dear. Is we can use a drug that specifically treats or blocks TMEM176 in our mast cells, and then we can induce them to degranulate, to activate. And when we do this, we actually see in the inflammatory mast cells that have, in which we've blocked TMEM176, they're not able to become inflamed to the same degree. So it seems that this marker is actually allowing them to contribute to the inflammation. So in this way, this is another way to perturb, to change the gene expression in these cell types. So this has gone a bit wonky, but that's okay. So how might we take this to the clinic then, as an example? So here we have our case study again. This patient, this is her treatment odyssey. But instead, what if she came in at diagnosis and we took a piece of her biopsy, a piece of her bowel, and we looked under a microscope and we saw that she had these inflammatory mast cells, as we can look on the microscope. And we treated that exactly. So we used a drug, such as the drug that we used in our petri dish, to turn off TMEM176 in these mast cells and stop their activated phenotype, stop their contribution to inflammation. We could achieve a situation where she's symptomatically well at the get-go, and that's guided by her individual biology. So that's really what we're hoping to achieve here, is by understanding the actual disease mechanisms that's happening in these patients and tailor treatment towards those disease mechanisms. So that's just a little snippet of what we do here at the Garvan, but I want to leave you with a few take-home messages from this little presentation. So firstly, the large bowel is a symbiotic environment, and it's really central to our general well-being, and it's really important to look after this. And when we get breakdown in the balance between our microbiome and the cells that exist in our bowel, that can contribute to diseases such as inflammatory bowel disease and colorectal cancer. And here at the Garvan, we really use genomics technologies to guide our understanding of what's happening in IBD in order to inform better ways to choose treatments and better ways to treat these patients. So that's all for me, and I will hand over to Viv again. Thank you very much again, and I look forward to the discussion.
Thank you so much, Kylie. That was a fascinating presentation. Just give us a couple of minutes while we get set up for our panelists. If I could now invite our wonderful panel to join us up on stage, please. So up on stage with me here today is also Dr. Jacqueline Turl, who works in the lab, in Dr. Kylie James's lab, has been working there since 2022, and now she is using something called single-cell RNA sequencing and computational techniques, which Kylie's already touched on. Bit of a mouthful, and hoping you can break down some of those techniques for us today. We also have Dr. Simon Garley from St Vincent's, who is a gastroenterologist and the clinical lead of the IBD service at St Vincent's. Thank you so much for joining us today. And we also have Madeline Jandura, who has ulcerative colitis, was diagnosed when she was a child, and we are just so thankful to have your perspective up here on stage today. Now, Simon, I was wondering if we could start with you. We've just heard a lot about the science of IBD. I was wondering if you could take us to the clinic and your experiments. What does the treatment look like from your perspective?
Yeah, look, I think the case that Kylie highlighted really high. It's one of our biggest challenges, and it's working out which treatment is the best treatment for which patient. And while we have some clues based on clinical factors, type of inflammatory bowel disease that may help us predict which treatment would be the best one to try first, it's still not very good. So our challenges really are that it's hard to pick which treatment will be the best one to use for which patient. We know that the first drug you choose is the most important one. That's the one that's most likely to work. So that early stage is so critical. Our other challenges with the drugs is that, at best we're looking at 60, 70% effectiveness. So we lack that next level of treatment that will really break through to be, 80, 90 percent effective. So it's really that, using, being able to personalize our treatment rather than having to fail, you know, treatments to find one that works. And these are usually 6, 12, 18 months, 2 years of, quality of life that's lost while, we're trying to find an effective, durable treatment.
So give us a little bit of insight into your work with Dr. Carly James.
Yeah, so it's been a really, fruitful collaboration, that we've had, and we're very lucky at St. Vincent's to have such a great institution at our doorstep where, I can chat to Kyle and talk about the problems that we have in clinic and the challenges that we have and where the clinical problems are. And then, she has, the fantastic expertise and technology to be able to investigate and look into these in more detail. And then we can work together to ask the right questions, and that will make a meaningful difference to patients and how we look after patients, not just here, but around the world. And then we can also help by engaging with our patients and getting the samples that Kylie needs.
One question, I guess, would be in terms of the research, how do you ensure that the findings, the markers that you're finding, um, have potential relevance in the clinic?
Yeah, so I guess it is a long process of, initially finding results, but then, testing them in, like I was saying, in on the bench and in animal models before we would then, look at taking that next step into human studies. So, this is generally a 5 to 10 year, if not longer, process.
But I think from the chart that we just saw, getting having that intervention early on can really take away from that long, arduous diagnostic odyssey. So the potential is extremely vast. Madeline, I was wondering if we could come to you next. Could you talk us through your personal experience with IBD, and could you potentially speak to what research, such as what Dr. Kylie James was talking about, could potentially mean?
Yeah, sure. I was diagnosed when I was 11 years old, and what sort of stands out is that you'd be able to tell me whether it was Crohn's or ulcerative colitis, which they were unable to diagnose me with for 5 years. And then it was just constantly in remission and then flare ups, different medications each time, lots of steroids, hospitalization. And it probably took over 12 years for me to really settle down and find what worked. So, yeah, this would really help in that initial stage with initial diagnosis, as well as finding sort of a treatment that does work and to stop, that quality of life being lost. Because that was really during my quite formative years of being a child and growing up in high school. I missed out on some of the HSC and uni and things like that. So, yeah, it definitely helped people with just that quality of life.
Because before a treatment is found, it's really about the diagnosis, isn't it? And you just mentioned it, that took some time.
Yeah, that definitely took a while. They were able to give me something which helped and settled it down, but it wasn't until we were able to really figure out what the diagnosis was that we could start trying all the different treatments. But, having just one would have been really amazing too.
Yeah, absolutely. And can you tell us a little bit more about how it's impacted your day to day?
I guess it is debilitating when it is a flare up. sort of you can't go out, you can't socialize, you can't work. You've sort of really got to be at home, close to that bathroom. As you know, you can't exercise, enjoy just like the general things in life that people do, I guess. It is very embarrassing, and it's hard to talk to other people about it or for them to understand what you're going through as well. I guess these days we are having a bit more openness, and you know, everyone's speaking about it and learning about it, but it can be quite hard, even just for a person to understand. Yeah.
I'm sure you would have preferred to be out doing the normal things that you would have wanted to be doing at that age.
Yeah,
You know,
definitely.
hanging out, heading out to the gym, things that were probably a bit tricky at that point.
Yeah, it was very hard. I mean, even just sort of going through puberty, I think it sort of stunted that a little bit as well. And yet going to the gym, just hanging out with friends, sort of just like discovering myself, I guess, like I was a teenager.
Kylie, I was wondering if we could come back to you now. What is your hope for your research? What do you hope the impact it will have?
Yeah, so as Simon said, it will take time. These discoveries always do to understand, the fundamental mechanisms underpinning these diseases. But yeah, the impact that we can have is profound. And I really, hearing Madeline talk, it almost brings you to tears. So yeah, just having kind of an impact on patients' lives, and even if we could just halve the time that it takes to get to an effective treatment, that's a lot of years across all of these IBD patients. So I really do hope that we can define what the different types of IBDs are at a mechanistic level, and then tailor the treatments to those mechanisms. That's what I envisage.
So, talk to us more about the diagnosis. You mentioned the different types. That's a really critical part of the equation, isn't it?
Definitely. So we've actually worked with Simon on a number of different types of IBD, including ulcerative colitis and the very rare form of IBD that co-presents with primary sclerosing cholangitis that I mentioned. And often they're kind of lumped together in terms of their clinical treatment, because although we can see that they're different in how they present, we don't have enough information to then treat them differently. So we compare basically the cellular mechanisms and the microbiome between these different patient groups to understand how they are actually different, to come to a more ground truth, I guess, difference between the different types of IBDs. Which, if we could come up with an assay, a set of markers that defines the different types of IBDs, then we can give a conclusive diagnosis at the start. So that's one element of something that we're trying to achieve.
Thank you. Jack, I was hoping we could come to you next. We just saw that beautiful abstract artwork on the screen there. Those, I think you mentioned there were 90,000 cells, 2.8 billion pieces of information. Absolutely incredible. Could you give us a little bit of an insight into your work at the bench? What does it take to generate this kind of data? Um, and how do you go about disentangling those 2.8 billion data points?
Yeah, sure. So actually, when it comes to generating the data, we do that very hand in hand with Simon. So that'll involve us actually going over to the hospital and collecting these biopsies on the day that the patients are there getting their colonoscopy. And then we have to work very fast after that. So we have to bring the biopsies back to the Garvan, and then we digest them using enzymes. And what we create is this single cell suspension. So we have all those different cell types there. And then we bring them up to the top level of the Garvan, and we run them through a microfluidics machine that captures each individual cell, along with all of the chemicals that are needed to sequence all of the genes in that cell. And then it gets sent off to sequencers at the Ramashotty, which is part of UNSW. A lot of complicated sequencing happens, and then what we get back is a really, really huge amount of data. And this then there's a lot of computational work that follows after that. which I won't go into the details of. But then once we see the cells, we start to define, we look manually at the sort of genes that are expressed. We can see the different cell types that are there. And then there's a lot of real literature searching and looking at what's already out there to help us decide about what we actually want to then pursue further. And that will also involve talking to Simon and clinicians and also thinking about what we already know about IBD and health. So, yeah.
One question from me is, when you come up with these discoveries, this Team M176, I think it was, is it sort of like a eureka moment? Is it a puzzle that's slowly coming together? I'd love for you to talk us through what a discovery like that looks like.
Yeah, I guess there is. So we could see that the cells are really different at first, and that was really exciting for us to see such a clear shift in the difference between cells. Um, but what we saw was these inflammatory mast cells that upregulate a whole host of genes that actually have no real known role in mast cell biology. So actually choosing which genes to look at a bit further is a lot of I guess looking at what's already out there, just like I said, but also looking at what's available. So what was really exciting is that when we were looking at all these genes, we found that there was actually already a drug on the market that actually targeted TMM176. So we thought that this was really exciting, but also what we found was that it was already associated with the development of colorectal cancer. So that also gave us a bit of a clue that it might be a good candidate to look at further. So that's where we kind of started with that. Um, yeah.
Sounds like a little bit, perhaps a little bit of eureka, a little bit of puzzle, and a lot of literature searching and getting into things. Simon, I guess where do the discoveries in the lab go in the clinic? What's the connection of feeding back into the clinic from the point of a discovery like TMM176?
Yeah, so normally once there's been a discovery like this, we then start thinking, well, what would be the next step in the research? And if it's an existing drug that's already available, then it would have been subject to appropriate studies for safety and things. And then you could go straight to designing a clinical trial. Looking at patients with inflammatory bowel disease and giving a new novel therapy, and comparing that to a placebo. Um, and so I guess there are different phases then of the clinical trial to looking at safety, looking at dosing, looking at its effect in a limited number of patients, and then finally the biggest studies, which are your phase 3 studies, looking at big multi-center, often international, multinational clinical trials, which is really to make sure and conclusively know that the drug is safe and effective before anything comes to market, and that we'd be recommending to patients.
Fantastic. And where do you see, from your perspective, IBD treatment going, say, over the next 5, 10, 20 years?
Yeah, look, I mean, it's been a really exciting journey. I mean, I've been treating patients with inflammatory bowel disease now for about 14 years, and when I first started, we literally had one biological drug available with infliximab, and just over the last 10 years, it's changed so much. So as a clinician, it's really exciting to have all this change and advances because, we can see the impact it has on people's lives. But we want to be able to do better. We don't want to try 5 drugs to find that the 6th one works. We want to avoid surgery if we can. We want to be able to tailor our treatment, not to have to do colonoscopies every year in every patient to pick up, the one bowel cancer out of like a needle in a haystack. So having genetic testing that can really tell you who are the most at-risk people to tailor everything that we do. So look, I think that's where the next 5 to 10 years is going to be, is really going to be discovering new therapies that are more effective, but also having the ability to tailor and personalize treatment, much as like what has happened in the cancer field, and being able to personalize treatment for patients with cancer. That is where we see happening in the field of inflammatory bowel disease. Mm-hmm.
Fantastic. Thank you so much, Simon. I might open the floor to questions at this point. Oh, we've got a couple of hands up already. Fantastic. Give us one moment while we grab some mics for you.
Kylie, great presentation. Thank you. CRISPR, seq-RNA is superseded CRISPR, I think. Is anyone using seq-RNA at the Garvan? Also, using other drugs or continuing to use monoclonal antibody drugs? And what other drugs are there on the horizon which supersede monoclonal antibodies? Thank you.
Interesting question. Kylie, would you like to
Your
take that one?
question. Yeah, so I think it's complementary technology, seq RNA. I don't know if anyone at Garvan is using. Oh, ask Jack as well because he may. Is it RNA seq, right? So yeah, RNA sequencing is kind of what we're doing with single cell sequencing. So the single cell technology is what we're doing here and what we've shown. So there's complementary in that it kind of addresses different questions. So rather than superseding CRISPR technology, CRISPR will allow you to edit the genome. Single cell sequencing or RNA sequencing will allow you to read out what those cells are. Yeah. And for your second question, maybe Simon's a better person for this.
Yeah, so look, in terms of the new therapies that are coming out, some are monoclonal antibodies, but there's been a big explosion now looking at small molecules, so tablet treatments. Ones, the ones called the Janus kinase inhibitors are tablets that are, again, probably one of the strongest therapies or most effective therapies that we've seen come out, and they've just made it onto the PBS in the last 2 or 3 years. There are other classes of small molecules as well as tablets, and so yeah, I wouldn't necessarily say that they are always more effective than the monoclonal antibodies, but they're definitely a different option for therapy if the others haven't worked, or sometimes we're using them up front. But still, our first drug, infliximab, is still one of our most effective treatments.
Thank you. I think infliximab, like vedolizumab, only lasts for 9 years. Are there any other drugs on the horizon? And is there a blood test that can determine whether you've got IBD?
So no, you're absolutely right that particularly with infliximab, the proportion of people that will lose a response each year, but it's not an absolute thing. I've still got patients, 15 years on, still on infliximab. And that's more the case with some drugs than others. Some have a more durable response. But yes, absolutely right that there is a rate of losing response that doesn't always happen. And so the second part of the question, were there a blood test for IBD? So the short answer is no. There are blood tests that are done to, maybe give you clues. Is it Crohn's or is it ulcerative colitis? But they're not very good. It'd be wonderful to have either a fecal or a blood diagnostic test. Yeah, which I think would have to be genome based. It would have to be a genetic sort of test. That's the only way I could imagine having a blood test that would be diagnostic.
Thank you so much. And could I please ask you to join me in just giving a big round of applause to all of our panelists today? Thank you.